In the last section, we discussed using physical methods of microbial control. And in this section, we're going to switch gears and look at chemicals. So we're going to be focusing on disinfectants and antiseptics. And we have to look at a lot of different factors to determine which would be the best chemical to use. Number one, we need to look at what microbe is being targeted. Are we looking at, viruses? Are we looking at bacteria? Are we looking at potential spore formers of bacteria? Are we looking at fungi? How clean does the item need to be? Are we looking at disinfection or are we looking at sterilization? What does the disinfectant do to the integrity of the surface? Is the disinfectant going to be so strong that it interferes with the integrity of the surface of the structure? And this is especially important to consider when we think about, IV tubes or catheters or things like that that could potentially be used inside living tissue. Is it safe? Does it put out noxious fumes? Does it cause skin irritation? So is it safe for us to use, and is it safe for the environment? Cost, of course, we have to consider, how expensive is it? And then how easy is it to use? So we have a lot of factors that we have to, discuss and look into before we decide on the correct chemical to use. In one of your earlier video assignments, you looked at Joseph Lister and how he began to use carbolic acid or phenol in surgery. He would use this as essentially a disinfectant for surgical wounds and was able to save a lot of people's lives that would have normally died in surgery. In 1879, Joseph Lawrence used Lister's work to develop Listerine, which of course we still use today as a mouthwash, and it has different disinfecting compounds in it. Today we don't use carbolic acid or phenol because it can be damaging to the skin. It can cause irritation. But we've modified the original phenol compound and call them phenolics. Okay, so they're similar to phenol, but we've been able to, modify the structure. So this is phenol right here. This is the chemical structure of phenol. And these two structures here that you see in B and C, these are phenolics. They've been essentially just modified. Some phenolics naturally occur, like eucalyptus is a naturally occurring phenolic. Other ones can be derived from creosote, which is a component of coal tar. It's what they put on railroad ties. Phenolics are pretty stable. They can stay on the shelf for a while. They don't immediately dry, so they can persist, in their wet form on a surface, which allows us to have an increased time of exposure. And of course, they're less toxic than phenol. They're not going to be as irritating to the skin. And they denature proteins and disrupt membranes, so they can act as a cytal agent to kill or an inhibiting agent, what we call a static agent. O-phenolphenol has been an ingredient in Lysol that we, use, Lysol wipes, Lysol spray. So that is a form of a phenolic. Hexachlorophene, or what we call hexadine, okay, or chlorohexadine, can be used on the skin. Triclosan was another phenolic. Of course, now that's pretty much been banned because we have a lot of strains of bacteria that are resistant to triclosan. And this little micro connections here talks about that. So heavy metals. Heavy metals would be things like silver or zinc, gold, and some of the first disinfectants that we used were heavy metals. They inhibit enzymes because the metals will bind to the proteins in the cells. And they are what we refer to as oligodynamic, meaning that we don't have to use heavy metals in high concentrations. We can use them in very small concentrations. And then the proteins denature. Now, one of the key points of using any type of chemical is this idea of selective toxicity. We want whatever chemical we're using to be harmful to the microbe without harming us. And that's the principle of selective toxicity. The problem with heavy metals, though, is that they are not selectively toxic to just microbial cells. They can accumulate in human cells or animal cells as well. So we have to be very careful when using heavy metals as disinfectants and antiseptics. There's a picture here in photo E down here, and this person is suffering from argyria, which is when too much silver has accumulated in the body, and it will turn the skin this blue-gray color here, and this is irreversible. Once this condition develops, it can't be undone. So what are some of the heavy metals potentially that we can use? Mercury. We've used mercury for many, many years. It was used to treat syphilis at first, but then we realized it could be used to treat a lot of different bacterial infections. But, of course, mercury is toxic. And I remember being a kid, there was mercurochrome and methylate, and both of these had mercury in them. Methylate was what we call a tincture, where we take a substance and dissolve it in alcohol. But I remember my grandmother using these. We called it monkey blood when we were kids because it was this bright red liquid. And, you'd get a cut. And I remember my grandmother coming in and she'd slather that stuff on there and it would burn like fire. And your skin would stay stained red for several days, but it never got infected. But now we realize, mercury is pretty toxic, and so these things really aren't sold in the United States anymore. You can probably still find it in some grandmother's medicine cabinet. Silver, we've used it for thousands of years as an antiseptic. Silvadene cream is used for burns. It has silver in it, and silver inhibits the growth of bacteria. We have silver nitrate, which can also be used as an antiseptic. And we can incorporate silver threads in linens, like, curtains in a hospital, or even bed linens. We can incorporate silver thread, and that helps to inhibit the growth of bacteria. We look at copper, nickel, and zinc, some others. Copper sulfate can be used as an algicide in aquariums. Metallic copper can also be used to inhibit microbial growth. Zinc chloride, zinc oxide can be used commercially. They're found in mouthwashes and antiseptic creams. So we are finding other uses for heavy metals, so it's possible that this list will grow in the future. So the halogens are those that are in group 17 of the periodic table. So we have fluorine, chlorine, bromine, and iodine. And we don't typically use bromine very often, but it can be used as an antimicrobial agent. Chlorine and fluorine are often not used on the skin, but iodine can be. It can be used as a tincture, mixed in alcohol. Or it can be used as what we call an iodophore, which is when we take iodine and mix it with an organic molecule like a sugar or fat or something like that, which increases its stability. One common iodophore is povidone iodine, which is commonly called betadine. That's the brand name. And so you see this image here of betadine being, spread on an individual's lower back here. So we use betadine in surgery because it can clean off the skin, and it's safe to clean off the skin. It produces very few reactions. Chlorine can be used for disinfection. Of course, we use it in water. This is what most municipal water supplies use for disinfecting their water. We can use, when you add chlorine gas to water, it forms hypochlorous acid. And this is what really produces that antimicrobial effect. Sodium hypochlorite, another chlorine-containing compound, is the main ingredient in bleach, and that's why bleach is such a good disinfecting agent. Chloramines are another form of chlorinated compounds. They are stable and can release chlorine over long periods of time. Again, used for drinking water. They can be in tablet form. So if you're, out hiking or you're camping, you can use these tablets to disinfect your water and for other needs. They are not typically used as antiseptics because they can irritate skin. Fluorine we use in its ion form, fluoride, and it's put in toothpaste. It's added to tap water. It's in mouthwashes as well. And it makes our teeth enamel more resistant to decay by acids that are produced by the bacteria that live on the teeth. Alcohols, of course, can be used as both disinfectants and antiseptics. They denature proteins and they denature cell membranes. Typically, a 70% alcohol solution is the formulation that we get, and it in fact works better than a 100% alcohol solution. And part of that is because of its evaporative properties. If you have very strong concentrations of alcohol, 95, 100%, it's going to evaporate so quickly that we don't get to reach that death time. We don't have enough time of exposure, so the microbes may not be sufficiently killed. But by having a 50, 60, 70% alcohol solution, then it's not going to evaporate as quickly. It can stay on the surface or even on the hands or somewhere on the body longer, and we don't have to worry about, did we meet that time of exposure? They can be bactericidal, so they can kill bacteria. They can be fungicidal, so they kill fungi. But they're not going to typically kill all viruses, typically only the enveloped viruses. Remember those enveloped viruses escape from the cell membrane and they take a piece of it as they go and kind of use it as this outer envelope. And since alcohol denatures proteins and disrupts membranes, it can cause issues with particular envelope viruses because it's going to break down that envelope. Alcohols, of course, are typically the main ingredient in hand sanitizers. Again, denaturing proteins, disrupting cell membrane, but they're not going to work as well in the presence of dirt. The dirt kind of inhibits the hand sanitizer from actually reaching the microbes that are living on the surface of the skin, especially if we're talking about our hands. We have all of those, crevices in there. And so bacteria like to get down in there, as you saw with your hand washing lab that you did earlier in the semester.